I think we all realize that drug discovery is a little bit in a challenge these days. But I have some good news, which I'll start with. And that is, despite actually kind of in the last several years, a rise in macromolecular antibody therapeutics, that the number of INDs last year accepted by the FDA, the majority were still small molecules. And I think this still indicates a very strong trend that small molecules are the cornerstone for drug discovery, despite maybe some of the challenges in terms of drug-drug interactions, specificity, things like that. I think the thing that we're realizing is that drug discovery is a multi-tiered problem. It's not just an issue of developing an active therapeutic against a target, it's also how you deliver that. How is it absorbed? How is it metabolized? What's its first pass pharmacokinetics? And then what happens downstream, for example, hERG interactions and other things like that that you have seen as challenges in the industry in the last several years. So we are understanding, I think, more the complexity of what we're trying to do in terms of developing effective therapeutics for disease treatment. And that not every one of us is the same, and that oftentimes we are working with a multi-tiered problem, whether it be pharmacogenomics or even just developing actives, as well as also being able to effectively deliver them as small molecule oral therapeutics. So I think we have a number of things. I think a lot of price challenges, but one of the points of this talk is to show you how ChemAxon, in this afternoon's sessions, how ChemAxon is really helping address some of these challenges in our industry. So we're getting a lot of price pressure, as mentioned, and we're seeing a lot of now, increasingly a lot of work being done by virtual or smaller companies or CROs, especially synthetic chemistry, outsourced services. We're seeing increasing a lot of interest in silico discovery tools for helping predict or understand what's going on as a diagnostic tool in conjunction with the wet laboratory work. And I think those are some things you'll see in some of the afternoon presentations today. And I think we're increasingly seeing a flood of data. I didn't want to use the word big data, but it is a lot of data. And it's not just internal data, but also external data, publications. How do people, for example, in oncology, keep up with the current trends in their own specific area? It's a flood. And so there's a lot of increasing interest in being able to do knowledge management to understand and pick up interest in those things. And we are also getting that area with our document destruction and also naming tools and helping mine data so that you can pick up the structures of interest in the context in the documents. And you've seen a little bit of this with our SharePoint technologies, and you'll hear more about this today. So, ChemAxon's technologies, and I've already summarized this a little bit, but we are helping you solve some of these issues. You heard a little bit about Plexus in terms of being collaborative, an interface for helping share data. That obviously goes hand in hand with our IJC web client, where you're seeing those become frameworks that people can use to help bring together data from disparate sources together in a common web platform. We'll talk today a little bit about Reactor for helping you predict actually how to do reactions and also eventually start being able to do reverse synthesis prediction. And we'll hear from Duri about Metabolizer and doing first pass P450 metabolism prediction, in addition to David Ding with our knowledge management suite, document to structure, and how you can start using that for actually pulling structures out of documents, being able to pull text around that. And eventually you'll hear more of this also in the context with other technologies we have, SharePoint, Marcouche, and so forth. So my point is, let's be part of the solution, not part of the problem. I used this slide several times, and for somehow it still resonates. But I also get the comment that I'm still on the lower side of this curve on the left, not the right. I don't know why. So I'm going to go briefly through the agenda and then flip this over, and we'll be on time.